you for joining us today for the webinar on Canaries in the Coal Mine, What We Can Learn from Aging from Survivors of Cancer or Childhood Cancer. My name is Jennifer, and I will be your WebEx host. Before we get started, I'd like to make a few comments before we begin. All lines have been muted upon entry and will remain muted for the duration of the webinar. Please submit your questions throughout the presentation in the chat or Q&A panel and select host from the drop-down. We will ask them on your behalf during the Q&A portion of the webinar. If you need to view live closed captioning, please refer to the link that will appear in the chat panel. I'd also like to note that this webinar is being recorded. And with that, I now pass it over to Jen Gita to introduce our speakers.
Welcome to the second installment of the Perspectives on Cancer and Aging Arti Hurria Memorial Webinar Series. Today's webinar is titled Canaries in the Coal Mine, What We Can Learn About Aging from Survivors of Childhood Cancer, and features Drs. Kirsten Ness and Gramatges. Dr. Kirsten Ness is a physical therapist and clinical epidemiologist and member of the faculty at St. Jude Children's Research Hospital. She has a BA in physical therapy, an MA in leadership, and an MPH and a PhD in epidemiology. She is a Catherine Worthingham Fellow of the American Physical Therapy Association and has been in physical therapy practice for over 30 years. Her research focuses on the observation and remediation of functional loss among persons who were treated for cancer during childhood. Dr. Gramatges is the research co-director of the Long-Term Survivor Program at Texas Children's Cancer and Hematology Centers, where she develops and facilitates local and national research that investigates the physical, medical, and psychosocial late effects of treatment for childhood cancer. Dr. Gramatges serves as chair of the Texas Children's Cancer and Hematology Center's Practice Standards Committee, and she also is an associate professor at Baylor College of Medicine, and her clinical interests include both leukemia and cancer survivorship. In today's webinar, first, Dr. Ness will review the aging phenotype observed in cancer survivors and related risk factors uncovered by studies conducted in survivor cohorts. Next, Dr. Gramatges will review the molecular and biological indicators of aging noted in survivors with an aging phenotype. Then, potential physical and pharmacological opportunities for intervention will be discussed. Lastly, and if time permits, we will take a few questions. The Q&A session will be facilitated by my colleague, Dr. Lisa Galicchio. If you have a question for Dr. Ness or Gramatges, you can type your question into the Q&A panel on the lower right side of your screen. So without further ado, I will now hand it over to Dr. Kirsten Ness. Dr. Ness, are you there?
I'm here, sorry.
There you go.
Our objectives for today are to describe frail health in childhood cancer survivors, to identify specific physiologic targets for intervention in childhood cancer survivors, to begin to understand the pathobiology of frail health in childhood cancer survivors, and to identify opportunities for prevention of frail health in childhood cancer survivors. So first, I'd like to define frailty, which is a loss of physiologic capacity that interferes with normal function. It's most commonly described in older adults. It can be distinguished from disability and comorbidity and identifies individuals highly vulnerable to adverse health outcomes. It often precedes chronic disease onset and is a predictor of early mortality. In the figure, the solid line represents normal physiologic aging. In childhood cancer survivors, we hypothesize that disease and treatment exposures change the trajectory of this line. And this is important because children don't have chronic diseases when they develop frailty and when they're treated for their cancer. And so they have a lot of information that might give us insight into aging in other populations, including adult cancer survivors. The trajectory of this line in childhood cancer survivors can be modified by lifestyle choices like sedentary behavior and poor diet. And I'll give you some examples of this going forward. Fortunately, this framework provides us with opportunities to design appropriate interventions for children with cancer and among cancer survivors, depending on where they are in relation to treatment and the development of adverse outcomes. So to test this hypothesis, we took advantage of the St. Jude Lifetime Cohort to describe physiologic frailty as a sign of accelerated aging among adult survivors of childhood cancer. In this cohort, we classified frailty using 5 components of fitness, first described among persons 65 to 100 years of age in the cardiovascular health study. These include muscle wasting, muscle weakness, self-reported exhaustion, slow walking speed, and low energy expenditure. Those with 3 or more components were classified as frail, and those with 2 components as pre-frail. Among the 1,922 survivors who were just over half male, a mean time from diagnosis of 25 years, and a mean age at diagnosis of 8 years, nearly 8% were frail and 22% pre-frail. This compares to percentages in the cardiovascular health study of 7.2 and 15%, and hinted to us that childhood cancer survivors may have an aging phenotype. Our survivors ranged in age from 18 to 50 years, whereas the individuals in the cardiovascular health study ranged in age from 65 to 101 years. As you can see from the yellow bars, frailty is not common in the healthy controls in our cohort, and pre-frailty is only prevalent among 6.2% of controls, similar in age to our survivor group. In our cohort, survivors are shown in the red bars. Low energy expenditure, low lean mass, and exhaustion were the most common components of the frailty phenotype. This table shows that in a median of 3 years of follow-up, frailty increased the risk of death in the St. Jude Life cohort by 2.6-fold, a rate comparable to that reported among older adults in the cardiovascular health study. In multivariable models, frailty was associated with cranial radiation, abdominal or pelvic radiation, and smoking among male survivors, but only with cranial radiation among female survivors. So to validate this phenotype, we looked at frailty in a larger cohort of childhood cancer survivors, the Childhood Cancer Survivor Study. Participants were nearly 11,000 members of the Childhood Cancer Survivor Study, which is a retrospective cohort study first funded in 1994. The CCSS includes 5-year survivors diagnosed with cancer when younger than age 21 years, between 1970 and 1999, and their siblings. It includes participants from 31 contributing centers across North America. Survivors with the most common pediatric cancers are included, and we have detailed treatment data as well as a different range of outcomes. In this self-report study, frailty was defined, again using 3 out of 5 of the criteria from the cardiovascular health study. Lean mass was defined as a BMI of less than 18.5 kilograms per meter squared, or having an unintentional weight loss of more than 10 pounds in the last year. Exhaustion was defined using the vitality subscale of the SS36, activity expenditure using the N-Haynes Physical Activity Questionnaire, and slow walking speed and weakness by self-report. The prevalence of frailty in this cohort was 3 times higher among survivors compared to siblings. Females reported a higher prevalence compared to male survivors, and the age-adjusted prevalence among survivors who were a mean age of 38 was higher than among those in the general population who were 50 years of age or older, and the prevalence of 50-year-olds in the general population is shown in the white boxes on the blue bars. In this cohort, when we looked at the association between treatment exposures and frailty, exposure to cumulative doses of cisplatin of 600 milligrams per meter squared or higher, cranial, abdominal, or pelvic radiation, lung surgery, and amputation were associated with increased risk for frailty. Importantly, when we adjust for lifestyle and grade 3 to 4 chronic conditions, only cranial radiation, pelvic radiation above 34 gray, and lung surgery were associated with increased risk for frailty, suggesting that even in the context of treatment exposures that can't be changed, attention to improving lifestyle and optimizing medical management of chronic health condition has the potential to prevent or at least delay the onset of frail health. As in older adults, the incidence of frailty in childhood cancer survivors is probably not static, but increases over time. In this next study, we evaluated the progression of frailty in survivors who were at least 10 years from diagnosis, treated at St. Jude Children's Research Hospital, and at the baseline evaluation were 18 to 45 years of age. They completed an assessment between 2008 and 2013 the first time, and then each participant returned to campus 4 to 6 years later for a follow-up visit. Participants were a median age of 7 years at diagnosis, just over half male, and 16% non-white. Over 50% of the cohort were survivors of leukemia or lymphoma. Of the 1,566 potentially eligible survivors, 1,432 returned for evaluation, while 77 died prior to recruitment. The prevalence of frailty more than doubled over 5 years of follow-up, while having 2 frail components, or being free from frails, increased only slightly. There were statistically significant changes in every component of the phenotype. Loss of function is evident, with substantial increases in those with slow walking speed, muscle weakness, and low weekly energy expenditure. There were 77 deaths in the cohort, and after adjusting for age, sex, non-white race, and chronic health conditions, the odds of death were 3.5 times higher among those who were frail compared to those who were not. Being frail conferred an even greater risk of death than did having a cardiac or pulmonary condition. Frailty is a fairly general construct, and so to identify more specific physiological markers to target with interventions, we set out to identify specific functional targets. In this study, we looked at exercise intolerance and muscular strength in the St. Jude lifetime cohort. Among 1,041 survivors, a mean age of 35 years, 64% of those exposed to cardiotoxic agents, which included anthracyclines and or chest radiation, and 56% of those unexposed had peak VO2 less than 85% of predicted. 30% of survivors had leg weakness. After a median follow-up of 4 years, there were 24 survivor deaths in this group, 21 among those with, and 3 among those without exercise intolerance, which equates to an adjusted hazard ratio of nearly 4, comparing those with to those without exercise intolerance. Unfortunately, as we show in this paper, functional impairments start early in children with cancer. Among 109 children with newly diagnosed ALL, 79% had impaired performance on the 6-minute walk test, and 30% had lower extremity weakness. Proximal muscular strength was impaired among these 109 children, and you can see how much different it is than population controls. And this is an example of what happens to muscle during treatment for T-cell leukemia. This is fat infiltration in a 12-year-old during maintenance therapy, which likely impacts his muscle strength. We also evaluated bone loss in children with ALL, and we can see that these problems tend to persist. You can see that bone loss is most prominent during treatment, and at week 120 when they, and to 2 years off therapy, it doesn't improve all that much. We also know that lifestyle is important, and this is a study where we looked for an intervention target, and we evaluated the association between dietary protein intake and lean muscle mass. As you can see in this adjusted model, we show that protein intake and participation in 1 or more resistance training sessions per week are associated with better lean muscle mass among ALL survivors. So before I pass it over to Dr. Gramatges, I would conclude that childhood cancer survivors demonstrate an aging phenotype, and that this phenotype progresses over time in young adults. Specific targets for intervention exist, for instance, aerobic capacity and muscle weakness. However, we need to look at some mechanistic studies that reveal new targets for intervention. And in the meantime, there are several interventions that are underway across the childhood cancer continuum. The next steps that we probably need to do are to continue to describe functional limitations in frail health and to understand the pathobiology and design and test new interventions that prevent or remediate frail health among childhood cancer survivors. And so now I will turn it over to Dr. Gramatges.
Okay. Thank you, Carrie. And for the second part of this presentation, we wanted to delve into a little bit more about the evidence for biological indicators and molecular mechanisms of premature accelerated aging, and how these indicators might differ or be similar between physiologic aging and the aging we see in survivors. So the objectives of this part of the talk are to review the hallmarks of physiologic aging, to review the current understanding of molecular mechanisms underlying premature aging in survivor populations, to discuss the potential impact of chemotherapy and radiation on general and organ-specific aging, suggest some opportunities for therapeutic intervention, and lastly, just to provide some considerations for anyone who might be developing a proposal that includes an aim that investigates aging-related biomarkers. So the risks for late effects in survivors are determined by treatment exposure and dose, and further influenced by demographics, behavioral and lifestyle choices, and genetic factors. These chronic health conditions that result from the accumulation of late effects over time and excess risk of frailty and limitations in physical performance, together, as Dr. Ness described, suggest that childhood cancer diagnosis and treatment augments risk for premature accelerated aging in survivors. So discussing each of these in turn, briefly, survivor risk for chronic health conditions we know now exceeds that of age-comparable controls, and this has been shown in the childhood cancer survivor study very elegantly, with an increased risk for specific chronic health conditions that are typically associated with elderly populations. And we know now that 1/3 of our survivors develop a severe disabling or life-threatening chronic health condition within 20 years of completing therapy. This risk has been mitigated somewhat with more modern chemotherapy and treatment efforts, but is still far exceeding that of sibling controls, and this is also from the childhood cancer survivor study. We also know, as Dr. Ness described, that survivors experience limitations in physical performance, such as decreased grip strength and slower walk speed compared with age-similar controls, and that survivors are more likely to be pre-frail and frail than age-comparable controls, approaching the frequency that is observed in individuals who are decades older. So the question that we had are the molecular mechanisms that underlie physiologic aging similar to those experienced by survivors with signs of premature accelerated aging? So in 2013, there was a nice review that was in Cell and enumerated 9 tentative hallmarks representing commonalities of aging across mammalian species. And these include those on the list: genomic instability, telomere attrition, epigenetic alterations, and loss of proteostasis will be discussed in this presentation, together with some correlative hallmarks that include mitochondrial dysfunction, cellular senescence, stem cell exhaustion. So taking each of these in turn, I'm going to discuss each of these hallmarks and then what the current evidence is available showing that these are also hallmarks in survivors. So with genomic instability, somatic mutations, chromosomal aneuploidies, and copy number variation increase with physiologic aging. We know that 10 to the 4th to 10 to the 5th DNA lesions are generated each day in our bodies, arising from intrinsic and extrinsic sources. And we also know that clonal hematopoiesis predicts all-cause mortality and risk for hematologic cancers, especially with physiologic aging in elderly individuals. So DNA repair mechanisms we have available to resolve specific types of DNA damage, like DNA mismatch repair, et cetera, but capacity for that repair declines with increasing age. There are specific aging-related clinical outcomes that have been associated with the genomic instability that include cancer, neurologic disease, osteoarthritis, and mitochondrial dysfunction, cellular senescence, and stem cell exhaustion. So what is the evidence in survivors? So there have been several studies now who have specifically looked at genotype-phenotype associations in survivor populations. One of these is a Children's Oncology Group study that has shown clinical and genetic risk prediction of subsequent CNS tumors in survivors of childhood cancer that specifically found variation in BRCA2, LIG4, and XRCC1, and associated with an increased risk for glioma and meningioma, in addition to variation in TP53. There's also been a study of neuroblastoma survivors that shows increased risk for second malignancies with variation that implicates DNA repair. And then another in Hodgkin survivors that show XRCC1 and glutathione S-transferase gene polymorphisms are related to susceptibility to radiotherapy-related second cancers in those survivors. And lastly, there's one very interesting study that looked at repair of ionizing radiation-induced DNA damage and risk of second cancer in survivors. And in this study, LCLs or lymphoblastic cell lines were established from cases with second malignant neoplasm and with only one history of cancer or first malignant neoplasm. Those lymphoblastic cell lines were irradiated, and the phosphorylated histone H2AX was measured in various intervals post-radiation exposure, showing that there was higher or increased evidence of early cellular response to DNA double-stranded breaks in those who had multiple cancer history. So telomere attrition is another of the primary hallmarks of physiologic aging. Telomeres are the DNA protein structures at chromosome ends. They shorten with DNA replication and with chronologic aging. And the loss of telomeric DNA with each cell division leads to replicative senescence or apoptosis, which is known as the Hayflick limit. There are a number of aging-related clinical outcomes that have been associated with excess telomere attrition, and those include cancer, pulmonary disease, atherosclerosis and coronary heart disease, osteoarthritis, all-cause mortality, poor grip strength, sarcopenia, and cellular senescence and stem cell exhaustion. There have been some evidence of telomere attrition or excess telomere attrition in survivors of childhood cancer. In one study of survivors of acute leukemia, there was evidence of shorter telomere length compared with controls, especially among survivors who received cranial radiation, which is no longer as common in the treatment of childhood leukemia, but is still employed for certain cases. We also were able to show a few years ago that telomere content was associated with risk for second malignant neoplasm in survivors of childhood cancer, an association that was predominantly with thyroid second cancer in survivors of all childhood cancers. And then there's a smaller study that had some interesting findings that found clinical and biological markers of premature aging after autologous transplant in neuroblastoma survivors who had significantly shorter telomere length compared with their age and sex-matched controls. Both of the two later studies employed a quantitative PCR methodology for telomere assessment, and the first study employed Southern blot. It should be noted that there are a variety of techniques that can be assessed to measure telomere length, and I'll talk about that in a little bit more detail. In the St. Jude Life cohort that Carrie described, there is also evidence of telomere attrition in survivors of childhood cancer. controls, survivors had much more pronounced telomere attrition with age than controls in that cohort, and also the mean lymphocyte telomere length was significantly shorter overall for survivors across all diagnosis groups when compared to controls that have no prior history of cancer. We have also been very interested in exploring differential telomere shortening and leukocyte subsets, and we have explored this in the childhood cancer survivor study that has a biobank with viably frozen leukocytes available for study. So we've shown that telomere shortening and leukocyte subsets with and without second cancers, there is pronounced telomere shortening in certain leukocyte subpopulations that include naive T cells and NK cells, more shortening than in other leukocyte populations, and that all survivors have shorter than expected telomere length compared with age-related norms. So in lymphocyte flow fish analyses, where telomere length is on the y-axis and age in years is on the x-axis, you can see the percentiles based on the age population norms that include the first to the 99th percentile indicated by color in the graph. And in survivors, all survivors had a deviation that was below the anticipated mean from the population, but there was a much higher also than expected frequency of telomere length in the less than first percentile in these survivors who were in their 40s, 30s, 40s, and 50s and shouldn't have telomere length that's below the first percentile in their lymphocytes. So these are probably impacts of therapy-related outcomes. Regarding epigenetic alteration, we know that DNA methylation is the process by which methyl groups are added to the 5' cysteine of CG dinucleotides, or CPGs. Epigenetic modification, such as global demethylation, increase in site-specific variability in DNA methylation occurs with aging. And the epigenetic clock is a technique that has been applied, and there are several different calculators, but essentially preferential hypermethylation of CpG island loci is used to correlate with chronologic age and develop outcome of the age acceleration residual, which is the difference between DNA methylation age and chronologic age. And you can see in the graph taken from one of these calculators from Nature Reviews that you would anticipate greater change in this age acceleration residual at younger ages that continues to progress over time, suggesting that DNA methylation age should correspond closely with chronologic age. Aging-related clinical outcomes associated with an increased age acceleration residual, so a DNA methylation age that exceeds the chronologic age, has been shown for elderly populations predominantly who are frail with poor grip strength, sarcopenia, cancer, and who have type 2 diabetes and neurologic disease. Epigenetic alterations have been explored in survivor populations. There have been studies looking at DNA methylation patterns of adult survivors of adolescent and young adults with Hodgkin's lymphoma that showed excess epigenetic aging, blood DNA methylation age in survivors compared with their twin, which is an interesting approach. There also has been evidence of DNA methylation association with obesity in survivors of pediatric acute lymphoblastic leukemia, which is a childhood cancer survivor study report. Essentially, BMI DNA methylation loci were associated with obesity in survivors who were treated without cranial radiation. There have also been studies of T-cell epigenetic remodeling and accelerated epigenetic aging and long-term immune alterations in survivors, showing accelerated epigenetic aging in T-cells specifically that were associated with chronic inflammatory disease. And then lastly, there has been a study of DNA methylation of a novel PAC4 locus that influenced ototoxicity susceptibility following cisplatin and radiation in pediatric embryonal tumors. We've also have some preliminary unpublished data in this area. In St. Jude Life, Dr. Ness has shown that the age acceleration residual is increased among survivors who were exposed to chest or abdominal radiation, which is in line with the prior published work, compared with survivors who were not exposed to radiation. This suggests that radiation may contribute to accelerated epigenetic aging. And in that study, the age acceleration residual was also associated or correlated with telomere length. In ALL survivors who were participants of the childhood cancer survivor study, we've also shown that DNA methylation age exceeds chronologic age by about 17 years. and that the age at diagnosis was inversely correlated with age acceleration, suggesting that younger subjects may be more susceptible to accelerated epigenetic aging. And then lastly, of the four primary hallmarks of physiologic aging, also is loss of proteostasis, where there hasn't been a lot of work in survivorship from childhood cancers, but I think could be an important area to explore. Essentially, proteolytic pathways that are responsible for the unfolded protein quality control become more hypofunctional with aging, and there are two systems that conduct this work, the autophagy lysosomal system and the ubiquitin proteasome system. Aging-related clinical outcomes that may be associated with impaired dysregulated proteostasis include Alzheimer's disease, Parkinson's disease, cataracts, and pulmonary disease. And this area is particularly potentially attractive because of the therapeutic druggable target. And there are a number of drugs that might be employed for this particular hallmark, but has not really shown, or at least very limited work in childhood cancer survivor populations. There are a number of other signs of aging that we could also discuss, particularly with respect to inflammation, which there have been a number of studies showing certain cytokines and CRP that is elevated in survivors compared with controls, in some cases after autologous transplant, and then also with some clinical outcomes that are typical of survivors, such as the impact of sleep, fatigue, and systemic inflammation on neurocognitive and behavioral outcomes in long-term survivors, where worse executive function, processing speed, and behavioral symptoms were observed in female survivors with higher levels of the noted cytokines and CRP. And also some evidence of elevated markers of inflammation and endothelial activation and increased monocyte counts in adult survivors of childhood leukemia, suggesting chronic inflammatory activation and immune dysregulation in adult survivors. With respect to oxidative stress and mitochondrial dysfunction, there have been some genotype-phenotype association studies in this area, polymorphisms in genes related to oxidative stress and inferior cognitive and behavioral outcomes in ALL survivors, and then also accelerated atherosclerosis, hypolipoproteinemia, and insulin resistance in survivors of Hodgkin's lymphoma, where markers of oxidative stress, including oxidation protein products, low-density lipoprotein, and CRP, as well as endothelial dysfunction, was higher among the Hodgkin lymphoma survivors compared with controls. And then also mitochondrial dysfunction. And so in the St. Jude lifetime cohort, and I should explain that altered mitochondrial copy number is involved in aging-related diseases, and a lower mitochondrial copy number has been observed in elderly populations. So in the St. Jude Life cohort, this was examined, and mitochondrial copy number declined with age at a faster rate than expected among survivors. So the loss was 9 copies per 10 years compared with an expected loss of 1.5 copies per 10 years, suggesting this also may be a feature of aging in survivors. With respect to cellular senescence, a number of years ago, one seminal study provided the first genetic evidence of a causative role of senescence in modulating aging and age-relating pathologies through a mouse model that carried a drug-inducible caspase that was driven by a P16 promoter. And with administration of a rapalog, a synthetic drug senolytic, the P16 positive senescent cells are removed from the system by apoptosis, and the effects in the mouse model included a reduction in aging-related pathologies such as lipodystrophy, glomerulosclerosis, heart hypertrophy, and cataracts, and also extended their lifespan. So this is beginning to be examined in survivors now. So muscle weakness, frailty, metabolic and cardiovascular dysfunction, as well as risk for second cancers, pulmonary fibrosis, and many conditions that are common to survivors are associated with pronounced cellular senescence in non-survivor populations. There are very limited studies in humans to date, but there is one that I thought worth discussing where the expression of the senescence marker P16, INK4A in skin biopsies of ALL survivors were almost 6 times higher in their scalp skin biopsies that were targeted by cranial radiation compared to biopsies taken from other regions in their body, suggesting that excess advanced cellular senescence may be predominant in survivor populations exposed to radiation. There are a number of potential interventions that we could leverage to target mechanisms of aging in survivors, and this is not meant to be a complete table, but to provide a framework for consideration of use of these agents in animal models and perhaps in human studies. I highlighted exercise and caloric restriction and intermittent fasting because those two areas are where we have the most data in human studies for effect in reducing frailty and improving aging-related outcomes in survivor populations, but certainly there are a number of other agents here with a great deal of potential that have been used to varying degrees in both animal models and in limited human studies, and that I think could be a nice platform for future clinical trial intervention studies. And then to conclude, and Carrie showed this graphical depiction a little earlier, but we believe that insults to molecular integrity and physiologic capacity provoked by exposure to chemotherapy and or radiation may be associated with an excess risk in advanced onset of age-related diseases and frailty in survivors of childhood cancer. But we know that physiologic aging is a complex, multifactorial process that is influenced by genetic, epigenetic, and environmental factors. And I think it's important to delineate the phenotypic differences between physiologic aging and the early aging experienced by survivors, because these are not yet clear, nor are the distinctions in the molecular mechanisms that drive these phenotypes. So there's a lot of work to be done just in the background, trying to set the stage for what are the differences and how, before we consider even more clinical intervention trials. Physiologic aging, it's important to realize, is characterized by disparities in functional decline between organ systems. So in general physiologic aging, there are relative rates of functional decline that differ between different organ systems. And that suggests there's inherent variation in tissue susceptibility to chronologic aging. And I think this highlights another potential difference in survivors, because chemotherapy and radiation may also differentially impact specific organ systems. So that this figure in survivors may look quite different and may actually be individualized depending on what each person has received as their treatment for their childhood cancer. So last, I just wanted to close with some final thoughts regarding grant development, just considerations and lessons learned for anyone who is developing a grant proposal in this area and would like to know from our experience, what roadblocks that we've had and concerns that we've had from our submissions. So I think it's, of course, first and foremost important to include the rigor of prior research. When you're proposing biomarker studies in survivor populations, you need to clearly delineate the evidence for association with aging-related outcomes in cancer survivor populations, and of course include any preliminary data that you might have in this area. I think it's important to consider molecular mechanisms that are associated with organ-specific outcomes. For example, if you're looking at cardiotoxicity, you would think about specific markers such as oxidative stress, where there's evidence and groundwork already that has been laid suggesting that reactive oxygen species impacts cardiotoxicity outcomes in survivor populations. One thing that we've gotten as feedback is that we really need to be careful about providing rationale for limiting investigation to a subset of biomarkers, because it's impossible to be all-inclusive, but I think it is possible to limit your list of what you want to investigate to a narrow number of biological factors. You just need to be able to provide rationale for why those were selected over others and recognize the limitation that it's not an all-inclusive approach. And also one difficulty that we've had in submitting grants in this area are that they're often reviewed by experts in geroscience who have come from a background of frailty and physiologic aging and may not be aware of or aren't necessarily fully on the same page that the similar phenotype exists in aging survivors versus an entirely different disease process. So I think it's important to acknowledge and anticipate potential differences between physiologic aging and survivor aging, and hopefully soon we can more clearly delineate what those differences are, and this includes both the phenotype as well as the underlying molecular mechanisms. And not that, a 40-year-old survivor is going to mimic the exact aging process that an 80-year-old is going through. but may actually have distinct and organ-specific differences. One thing that I think is beneficial if you have access to biobanked samples is to consider the relationship of your outcome with longitudinal changes in biomarkers over time, because I think that's a much more powerful indicator of the impact of these molecular mechanisms or biomarkers in aging if you can show a trajectory over time versus capture only one point in a cross-sectional study. And then lastly, considerations, of course, of cost, feasibility, and power. DNA methylation or microarrays are expensive, and so is lymphocyte telomere length. There are alternate methodologies that may be less costly, and depending on how many biomarkers you're investigating and how, in your grant budget, there may have to be some considerations of those factors and selecting ones that are more cost efficient. And that is my last slide. So with that, I think we can have some time for questions.
Great. Thank you both for such a wonderful presentation. As a reminder to our attendees, you can submit your questions using the chat and Q&A box. Please submit them to the host, and I will help facilitate those over to our moderator. Lisa, take it away.
Yeah, thanks to both of you for excellent presentations, a lot of information. The first question is for Monica. You talked a lot about evidence in childhood cancer survivors. Is there any evidence for changes in these biomarkers that you described in adult cancer survivors?
So yes, I really highlighted and focused on the childhood cancer survivor population for this presentation, but certainly, and as is the case in many survivorship research studies, there is often more evidence and stronger evidence in adult survivors than there of adult onset cancers like breast cancer because of the sheer sample size and numbers and prevalence of those cancers. So I did not highlight those. But yes, certainly there are in the areas that I described, studies in survivors of women who had breast cancer, for example, and telomere attrition, DNA methylation changes there, and that's something you could certainly find by literature review, but certainly that is the case.
So here's the million-dollar question for you, Monica. If a researcher is doing a study and they want to include one or two aging biomarkers, can you give a recommendation, or does it depend on the research question?
So I can tell a personal story about this because I have not yet been successful in this in this area. I can tell you from personal experience that I submitted an R01 that initially selected three specific biomarkers. The response from reviewers was why I had excluded a number of other biomarkers. The reviewers wanted us to be more comprehensive. Then we resubmitted the grant with a more comprehensive approach and were told that we were being too broad. So I think, I don't think there's a great answer for this, and that's why I say for the next and hopefully final submission of this proposal, my plan is to very clearly focus on the areas where I have the strongest preliminary data. There are, for example, for certain of these biomarkers, there are meta-analyses that are consistently show associations with outcomes in elderly populations that I think we can say are the most likely to be associated with aging and cancer survivors, and then try to strengthen the preliminary data that we're able to present in our survivor population, showing that these biomarkers will in fact also, we have strong scientific rationale to propose that they will also be important in survivor populations. And I think that's really the key, because it's hard to be responsive to reviewer comments that are different, but I think that has been our general plan to proceed with where there's the most data and consistent data and where we have the strongest preliminary data.
Thanks. Here's a question for Kiri. Have you conducted exercise interventions among cancer survivors, and have you seen effects to minimize or mitigate some of these adverse health outcomes or changes in these biomarkers?
So we've done a resistance training intervention in childhood cancer survivors who are young adults. And, you know, based on the data that I showed today, we supplemented their protein intake, thinking that that would, as it does in older populations, enhance, or in athletes, enhance the amount of muscle gain. And we got a significant change in muscle strength in both the group that got the protein supplement and the group that got the placebo. The group that got the protein supplement had a small but not significantly different than the other group change in lean mass, you know, a little bit more. So we've tried that. I think that we didn't measure any of these aging biomarkers in that study. We just looked at lean muscle mass. And so in the future, we should look at that. We have an ongoing study that is doing an intervention in children right after they get done with cancer therapy, and we are looking at some biomarkers of cardiometabolic health, including some inflammation, but we're not looking at specific, you know, methylation or telomere length in that young population. And I think that what's probably really important is the studies that Monica is talking about in terms of, you know, understanding which biomarkers are important in childhood cancer survivors has to, you know, happen first, because we have to know that, in fact, they have things that the biomarkers will appear, and that they have biomarkers that differ from, say, healthy controls, so that we know that if we do an intervention, we can use that biomarker as an outcome before they get frail. Because I think the ultimate goal of any sort of an exercise intervention for children with cancer or for survivors is to prevent them from becoming frail in the first place and not have to use the frailty measures as an outcome. So I think that the biomarker understanding is really essential to this research.
Thank you. The final question that I have, unless some of our participants on the webinar submit questions, which we encourage, we have time. In your observational data, have you seen that poor diet and smoking and some of these adverse behaviors amplify the adverse effects of treatment on frailty? So if they have multiple risk factors, is it worse?
Yes, so in our study where we looked at frailty in the St. Jude Life cohort, we identified smoking as a problem for males, and in the CCSS cohort, and I didn't show a lot of this behavioral data today, we identified you know, sedentary behavior as a risk factor for frailty. And then in our study where we looked at exercise intolerance, we found that smoking had an impact on exercise tolerance, as you would expect, but that also physical activity was a strong predictor of, physical inactivity was a strong predictor of having, you know, poor exercise tolerance and having frail health. You know, in the general population, we know that health behaviors are extremely important, and I think that that's also true for childhood cancer survivors, or for any cancer survivors. And one of the things that we need to do when we do physical activity and or exercise interventions for this population is really treat them like they're somebody who needs rehab, because they have some impairments at the end of their cancer therapy that make it maybe more difficult for them to successfully participate in physical activity or follow a caloric restriction diet, which is, you know, certainly a way to prevent aging. But we have to make the intervention doable and allow for people to be successful when they start moving and or dieting or quitting smoking, so that they continue with this, you know, optimal behavior over their lifetime.
I agree with that, Carrie, and I would also add that there are a number of studies that have shown that survivors of childhood cancer are very sedentary, have a much more pronounced rate of a sedentary lifestyle than those who aren't survivors. So why is that? And I think we need to think about contextualizing frailty in survivors and consider the other outcomes that survivors are at risk for. For example, 1 in 5 survivors of childhood cancer suffers from chronic pain. And how does that impact their risk for premature accelerated aging? Is that also affecting their risk for sedentary lifestyle, and is that accelerating further their aging trajectory? Chronic stress is another factor a lot of our survivors struggle with for various reasons. They are much more likely to not get married, to not have a full-time job, to not complete education, to have insurance issues. How do all those factors also further contribute to the aging phenotype? I think this is a multifactorial problem that needs to be considered really in the context of what the outcomes that are specific to survivors. These are people in 30s, 40s, and 50s. These aren't like 80-year-olds. So they're experiencing a lot of external stressors and additional outcomes that are not necessarily characteristic of people in their age group.
And a related question that we just got is on light or moderate alcohol consumption and whether you have any data that you've looked at to see how that relates to aging or these aging outcomes?
So we have a paper that just got accepted in Clinical Cancer Research today that shows the impact of lifestyle on telomere length. And as in the general population, moderate alcohol consumption versus being a teetotaler or too much alcohol consumption is protective for telomere.
How do you define moderate, Carrie?
So drinking less than 7 drinks a week, but still participating.
Oh.
For women, men get to drink more. But, yeah, so they're bigger.
And one last question is about successful aging. A lot of the graphs showed worse aging among childhood cancer survivors. Are there any groups of cancer survivors, either childhood cancer survivors or adult cancer survivors, where there are data that there may be more successful aging?
That's a good question. I don't have. I mean, there are different degrees of risk, but not that I'm aware of that any group is particularly successful at it. I think from my perspective, it looks like everyone's not great at it, but some people are worse at it than others. I don't know, Carrie, what would you say?
Yeah, I would say that's true. And I would also suggest that if there are investigators or mentees that are interested in looking at that question, that the Childhood Cancer Survivor Study, I'm going to advertise now for NCI, is a resource for research, and that the data are available for use, and that the St. Jude Lifetime Cohort data are posted on the cloud at St. Jude and available for public use by anybody. And so if you're interested in that, you can drop me an email and I can refer you to the appropriate spot.
Great. And I think that's all the questions we have. And so again, I want to thank everybody who's been participating on the webinar for joining us, and also Kiri and Monica for their great presentations. Our next webinar as part of this series will be on April 9th from 12 to 1, and it will feature Luigi Ferrucci and Morgan Levine. And we hope that everybody will join us for that webinar. Thank you.
Thank you.
Thank you.
Thanks, everybody, and thank you again to all of our participants for your participation in today's webinar. A recording will be available within the coming week. Please be sure to check out the BRP website, but more information will be sent once available. Thank you again, and have a great day.
Thank you.